So this is a complete switch in gears. And I first want to thank Shitabra, who I don't see around, maybe he's, oh, there he is, for very kindly inviting me. It's been really very nice for me to be here to listen to things which I don't, usually read a great deal about or think about. And I hope the cell biology that we do will be of some interest to this group. So the general area of research that I work in is in regulation of synaptic vesicle transport. Yesterday, Krishanu very nicely introduced the problem to everybody in this group, and today we've had two speakers who use the model that I use, which is C. elegans, who've already introduced the systems and its advantages. But I'll give you a very, very brief overview of the question and what has motivated our studies so far. So the process of axonal transport is one which carries multiple cargoes from the cell body to the synapse and from the synapse back to the cell body. In the anterograde direction, multiple cargoes such as synaptic vesicles and mitochondria move from the cell body to the synapse, and the reverse direction is called retrograde transport. Axonal transport is necessary in all neurons. It's fundamental for the formation and function of the nerve system of which the brain is part. And defects in axonal transport have been implicated in either initiating or in the progression of several neurodegenerative diseases. One example which Krishanu gave yesterday was ALS and Alzheimer's. It's also intrinsically interesting because if you think of flow of cargo and you think of the distances that need to be covered, I'll give you a very simple example. If you look at a 0.5 millimeter neuron, and you have a vesicle which is about 60 nanometers in diameter, given cellular viscosity, it would take 6 days to get from the cell body to the synapse. And obviously, this is a really long time when you look at the pools of synaptic vesicles that are known to be present at the synapse. So you need efficient modes which are active, use energy to move cargo along this path. So how does the cell decide to do this? And in fact, why do you even need to do that? One needs to do efficient axonal transport because the majority of the protein synthesis and cargo biogenesis actually takes place in the cell body. So cargoes that need to move in the axon need to be able to distinguish point A, which is the cell body, and point B, which is the synapse. And the way the cell has solved the problem is to use a two-part polarized system. The first part of it is the roads. The roads, which are microtubules, are arranged such that the plus end of the microtubules face the synapse, the plus end being the faster growing end and not the charge. And the second part comes from molecular motors. For instance, in the anterograde direction, it's the kinesin family, which uses ATP and walks hand over hand towards the plus end of the microtubule using energy. In the retrograde direction, it is largely the dynein motor, which carries cargo away from the synapse so that you have very constant and good flow of material going in both directions, maintaining homeostasis at the synapse. So although we work on retrograde transport as well as transport of mitochondria, those are smaller efforts on our part. For today, I will focus on this one essential cargo, namely synaptic vesicles. It's essential, it's abundant in neurons, and it's critical for neuronal communication to work. The energy-consuming molecular motor kinesin 3 is essential for transporting the motor from the cell body to the synapse. Upon removal of the kinesin 3 motor, you can look at C. elegans, Drosophila, anecdotal evidence from zebrafish, mouse, or human. What happens is you lose synaptic vesicles at synapses, and they often accumulate in the cell body. So it's sort of almost like a precursor product relationship. And animals that lack these vesicles at synapses essentially are unable to move, breathe, eat, and often die. So it's obviously a critical and major regulator of synaptic vesicle transport. So let me take a step back and sort of describe the process as the field thinks about it. Okay? So anterograde synaptic vesicle transport is a complex process. you obviously need to make some kind of transport carriers which contain multiple synaptic vesicle proteins. This transport carrier needs to recruit the motor, often thought to occur at the cell body, but can also occur in other places. This needs to be directed towards the axon. You don't want this cargo motor complex to enter the dendrite. And once it's in the axon, you need to have processive motion which covers long distances. Remember, yesterday Krishanu mentioned longer motor neurons. The longest motor neuron in you and me is a meter long. It starts in the spinal cord, goes all the way to the big toe. Right? So you need very efficient processive motion. You walk along microtubules. You probably fall off the microtubules, get back on. You need to recognize you've reached the synapse. Usually there's a shift between the microtubule cytoskeleton and the actin cytoskeleton. You release your cargo, and then the free motor needs to be handled both at the synapse and the cell body. If you look at the kinesin 3 pathway in the field as such, what's known about it, there are several instances of regulations that have been published by various labs. For instance, one molecule, CYT2, initially described by Mei Zhen, and we still go back to her paper to read it, has been shown to be important for regulating motor processivity. R8, shown by Kang Shen, is supposed to be important in the early part of the axon in maturation, preventing aggregation basically of vesicles. And our own lab has discovered few new instances of regulation, which I shall describe today. So the major goals of my lab are to find molecules and mechanisms governing these multiple steps. And a longer-term goal is to relate these mechanisms to the biology of the organism, for instance, for synaptic development or function. And we've taken only very tiny baby steps in that direction, and there's also a couple of posters here for efforts that we are attempting. And we would appreciate your feedback. So today I'll give you a bird's eye view of our research, starting from the cell body. So as a geneticist, if you really think about the problem of what moves synaptic vesicles in the axon, a very simple approach is to use a genetic screen, much like what Kathy Rankin described, wherein, in our case, we would look for vesicles that are not present in synapses but are stuck in the cell body, like you see with the motor. Such screens have been done by the Jin lab, the Bargman lab, the Nonnet lab, and of course much more recently and much more extensively by the Kang Shen lab. And they were all generally to understand the problem of synapse formation. And none of those screens pulled out really significant, or even, I'm going to argue, very many, wherein you saw very strong accumulation of cargo in the cell body, which is that if you remove the kinesin 3 motor, you don't see vesicles at the synapses anymore, and instead they accumulate in the cell body. So when I started my lab, what it suggested to me was that the kinesin 3 motor is obviously a major requirement for synaptic vesicle transport, but it also suggests that there are other regulators, and there are other reasons to think that, but these might have more subtle phenotypic effects. So in a situation where the regulator by itself may not have a strong action, its role might be much more readily apparent in a sensitized genetic background, and it's a very standard genetic approach that people use. So what my lab did was we took a strong kinesin 3 allele that we showed was defective in attaching the motor, which could not attach to the cargo very efficiently, and then fed the animal a mutagen to drive now synaptic vesicle proteins back into the axon. And these double mutants would have cargo in the axon, and such a screen, we reasoned, would have the potential to identify either novel molecules or novel functions of known molecules that play specific roles in the context of synaptic vesicle transport. So what might have we expected to get from such a screen? For instance, I said we started with a motor which was not able to attach to cargo. We could have expected compensatory changes which might allow the motor now to attach to cargo. We could have also have activated other kind of molecular motor systems which could maybe compensate for the lack of the major motor. And of course, last but not the least, you could just be now putting synaptic vesicle proteins in different compartments and getting them out to the synapse in a completely different way. So we cloned one of our suppressors, and we need to work on some of others, and we found that was a molecule called JIP3, or junk interacting protein 3, or UNC16. And that was a very unhappy day in the lab when we found that out. And it was unhappy because this is a molecule that had initially been discovered by Roger Davis's lab as scaffolding stress kinases. It had been shown by Larry Goldstein's lab and also in Yishi Jin's lab. Subsequently, was an adapter for the kinesin 1 motor, which is a completely different motor. And then again from Larry Goldstein's lab, that it interacted with a retrograde motor. So it seemed like an embarrassment of riches, a molecule which does so many things. We pulled up in our screen. And in addition, Yishi Jin had already reported in her 2001 study that this molecule could indeed suppress the phenotype of the motor. But my student Vikas, not only talented but very persistent, kept at the problem saying, okay, let's try to understand what is going on. So the first thing, and if you remember the 3 ways we could have gotten, what kinds of processes we could affect in our screen, we said, let's look at the first thing. Are the phenotypes dependent on the action of the kinesin 3 motor? So we removed the kinesin 3 motor completely. All the cargo is stuck in the cell body. Animals essentially can't live beyond this stage. And then we removed the GIP3 protein, and we could see that the cargo, synaptic vesicle proteins, were entering into the axon. So our first hypothesis, that whatever we get as a molecule in the screen could be attaching the poorly functional motor on the cargo, was wrong. We did a lot of experiments, which I won't go to today, and looking at motor compensation by these known motors as well as others, and we didn't find any. So like Sherlock Holmes, when all the obvious has been ruled out, the only thing that's left is something has to be the truth. So we just looked at it and said, we must have some kind of role in making cargo. Okay? And even though I'm taking you on this detour, we'll come straight back to transport. So just bear with me. So what I'm going to show you here, and he actually caught it first by doing these kinds of imaging analysis. What I'm going to show you is a wild-type neuron where we can see synaptic vesicle transport in a living animal. You will see cargos that are moving in both directions as well as cargos that are stationary. Right? So you can see that very nicely moving. So antegrade, retrograde, stationary ones. Now just see what we see when we look at the GIP3 mutant. You see something big come out in the cell body. You see another one. Took because many, many hours of imaging, that's a scientific one, and then you have this big tube that comes out. So this was really exciting because this suggested that there was something which was not motor dependent but occurring in the cell body, wherein we were not able to package our cargo into the right compartment. And in fact, we went on to do the correct genetic experiments and see that the synaptic vesicle proteins can come out into the axon independent of the kinesin 3 motor, but independent from the molecule that had been published by Kang Shen as being very important for synaptic vesicle maturation. So in our late mutants, cargo is stuck pretty much in and near the cell body, but when you remove our late as well as you mess up the process which GIP3 is involved in, you can put cargo all the way out, including up to the synapses. So when we saw that the defect was in the cell body, we went back and said, there must be, GIP3 must be in the cell body, and perhaps it's localized to compartments which, are important in sorting and helping making the right kind of cargo compartment. And that's indeed what Vikas found. He found that GIP3 co-localized with a Golgi marker, which was mannose 2. So that was consistent with our observations about defects originating in the cell body. And once we found that, we said, is it only messed up in one way, or are there a lot of problems with it? And in fact, I'll just show one of the phenotypes that we found, which is that when you look at resident Golgi enzymes, usually they're restricted to the cell body in the neuron, but they now end up coming out all the way into the axon. And in fact, in some animals, you will see that the Golgi, there are many more puncta present in the cell body. So clearly, it suggested to all of our burden of evidence, putting resident Golgi enzymes into the axon, we're putting dendritic proteins into the axons, we're putting axonal proteins both, in aberrant carriers but also into the dendrite, that there were widespread trafficking defects in this mutant. And this was the basis of suppression of the kinesin 3 mutant. At this point in time, we felt that we really needed to understand what was going on and why we were seeing these kinds of effects and why GIP3 was doing what it was. So we took advantage of the fact that several proteins had been reported to be things that messed up the polarity of the neuron based on the fact that synaptic vesicle proteins, which were destined for the axon, ended up in the dendrite. We screened through a large number of, not large, there were only about 3 or 4. We screened through them, initially, and we identified that LARC, which is a Parkinsonian kinase, shared phenotypes with GIP3. What did it share? It shared the fact that it could make large cargo compartments, much like GIP3. It put dendritic proteins, it put axonal proteins into dendrites, but it did not put dendritic proteins into axons. But resident Golgi enzymes were able to come out into the axon. In every case we looked at, LARC had similar phenotypes to GIP3, most cases, except the dendritic proteins, but was less severe. And double mutants looked like GIP3 mutants. So after we saw that, we said, what happens? This is a kinase which had already been reported by Naoki Isamoto's group, was present on the Golgi. So can you now control what goes into a cargo vesicle by putting a lot of LARC in the cell when it lacks GIP3? And this was these experiments. What we saw was that when we overexpressed LARC, we could suppress the mistrafficking defect, as well as how many large transport carriers were formed. In addition, using transgenic lines that had been provided to us by Naoki Isamoto, which rescued the phenotypes, we were able to show that LARC and GIP3 were present in the same complex in a worm. And last but not the least, we saw that in GIP3 mutants, the LARC localization was changed so that it was no longer tightly localized to the Golgi, but seems to be slightly more diffuse. And another component of, which was very important for sorting on the cargo complex, which was ANC101 or AP2, was also mislocalized in GIP3 mutants. So putting all of this together, this is the conclusion that we came to. I'm sorry I'm cutting the story short. For those of you who might be interested, we can discuss it later. We think that GIP3 and LARC2 may be present on the Golgi or on some endosomal structure, which is important for sorting, acts as a negative regulator, prevents molecules from being put in the wrong compartment. When you lack GIP3, you're not able to recruit LARC2 effectively to the Golgi. Neither are you able to recruit the sorting molecule AP2, and therefore now you lose your molecular fence and things sort of get into compartments that they shouldn't. These compartments are moved by other motors, not just kinesin 3, and therefore they're able to enter the axon as well as the dendrite, and we see the suppression. When you remove LARC2, the phenotype is not as extreme. You do have some errors, but it's not as extreme. So what? This is an advanced test on the problem of axonal transport. In fact, this was through a long series of conversations that me and Vikas had. We said, and I kept telling Vikas, this is very important because what we have found is that for the first time we have found something where you have cargo being formed, and we have an insight, we can probably gain some kind of signature as to what is happening to the motor. Because if you don't make the right kind of cargo, the motor probably has less surfaces to be recruited to. And I've been looking for this kind of signature for a very long time. So Vikas then went on and I will skip this slide, went on and looked at what was happening to the synaptic vesicle motor, and he saw the following. This was the most exciting finding for us from this study, or at least for me. In wild type, a lot of the motor is in the axon. There's very little present in the cell body. But when you look at the GIP3 mutant where you're not making the right cargo carrier, you find that the motor accumulates in the cell body. In LARC, where most of the trafficking is more or less normal, you don't have as much accumulation, and if you overexpress LARC, you can suppress the higher levels of the motor that end up in the cell body. So what we learned from the study is that GIP3 is very important for the correct trafficking of synaptic proteins, and we think it occurs from the cell body, possibly the Golgi or the endosome, and it acts along with LARC1. But what is much more important is that a correct transport carrier is necessary to be able to recruit the motor. And in addition, what we gained for our future studies, which we are already finding pretty useful, is that the motor accumulation in the cell body is an additional marker for defective transport, where possibly you're not recruiting the motor onto the cargo surface. So after I've gone through all the genetics, it's probably harder, we might not be quite fit with this group. I'll come to something which I'm very fond of, briefly touch upon that. So when the motor cargo complex leaves the cell body and comes into the axon, you of course have motion taking place, right? You have, as you saw in some of the movies that Krishanu showed, that you can have anterograde and retrograde motion as well as the movies that I showed. So when I chose worms as a model system to start studying this problem, one of the things which attracted me was actually the touch neurons, which is very, very well characterized from its ultrastructural level. So you know the number of microtubules, you know the length of microtubules, and this is all very, very important, although I'm not going to show you in what ways it was important for our study. So the next thing was we started doing imaging systematically. We figured out that we were getting a lot of artifacts when we used anesthetics, so we developed a microfluidic device to do imaging. My postdoc Sudeep Mondal did that, and these were our collaborators. Kaustubh, who kept encouraging us in the beginning, saying we could do it and it was easy to try. And let me just show you our efforts in this direction. All right, so what you're going to see is a little worm which is moving around. You trap it, and I want you to note a few features of the transport. Already seen that. You'll see vesicles moving in the retrograde direction, vesicles moving in the anterograde direction, and you see stationary cargo. Okay? And you'll see that stationary cargo begins to break up. You'll find sometimes vesicles can go past stationary cargos and sometimes they cannot. And if you sort of think about the part that this cargo needs to take, it's an incredibly crowded environment. It's filled with microtubules. There are very little spaces where a vesicle can actually squeeze through, and that's we can leave that to discussion as to how that happens. So it's a neuronal highway filled with microtubules. And we're actually looking at, because we got so much quantitative data, working closely with Gautam and Vinod, his JRF here, we developed a quantitative model to sort of explain the various motions that we came up with, and we can discuss that later if someone is interested. But it was clear, most people look at moving cargo. We decided to look at stationary cargo because 40% of the neuron is covered with stationary cargo. So it seems like something we should be looking at carefully. And these stationary cargos can be long-lived and can last in a particular location for up to 3 or 4 minutes, and often reform at the same location even after dissolution. So we asked a very simple question. If you have a traffic jam, like you might have seen on Indian roads, does it influence the motion of moving cargo? It's a very simple question. And we in fact found that it did. We found that there was much more effects on the anterogradely, that is going towards the synapse motion, and less in the retrograde direction. So these are some experiments where we bleach stationary cargos and track every single vesicle that comes through. You can see a retrogradely moving vesicle actually moves past the stationary cargo, and an anterogradely moving vesicle stops. And if you look at the numbers that come from such experiments, you find that over 50% of the vesicles actually stop when we do this kind of careful analysis. And in the retrograde pathway, in the retrogradely moving vesicles, only 15% of the vesicles stop. In fact, this is a characteristic not only of the touch neurons. You can look at other neurons like motor neurons. We see the same thing. We look at dendritic cargo like glutamate receptors. Any cargo that is moved by the dynein motor, we find is able to blow through a stationary cargo. And this is because the dynein motor is more versatile. When it walks on the microtubule, it can walk backwards, it can sidestep, which allows it, like the Indian auto rickshaw, to bypass a traffic jam, unlike a big truck, which would have a harder time doing that. Okay? So essentially, an anterogradely moving vesicles came frequently to a stop. And this is not unique to the C. elegans system. So Sudeep actually built an additional microfluidic device to do imaging in Drosophila, and Shikhar did those movies. And what we could see was that we saw the same kind of differential when we looked at synaptic vesicle markers. Anterogradely moving vesicles stopped much more than retrogradely moving vesicles. And we haven't looked at 90 mutants here, but we really should and can do so. So, we did all the cell biology, and then the question was, why should you care? So what if you have stationary cargo vesicles? Maybe it's just that the motors fall off the microtubules, motors fall off the cargo, or you come to the end of a microtubule, or there's a traffic jam. So the question in other ways is, do stationary cargo have a role in the biology of the neuron? And this is where it was kind of nice that we were able to come this far. What we asked was a very simple question. So Kausalya, who used to be a postdoc in my lab, he said, let us just stimulate the animal repeatedly. We initially started with 5 stimulations and nothing happened. So we stimulate the animal repeatedly using gentle touch and look at cargo transport in the touch neuron. So what would we see? When we stimulate the animal 30 to 40 times using an eyelash, and then we image, here are multiple chymographs, what we find is that the number of stationary cargoes drop. And I'll show you a later figure where we see the number of vesicles per stationary cargo drop. And in about an hour after the stimulation, it comes back up. So we did it this way, and we also had this artificial dirt-type device that Sudeep made, and we just let the animal bang against these pillars. In this case, we don't have very good control over how many stimulations it undergoes. But even in this case, we see that number of stationary cargo drop and amount of signal per stationary cargo drops. So if we actually look at every stationary cargo, they have a large number of vesicles. After repeated stimulations, you lose all the higher intensities. In fact, the vesicles are much fewer at those stationary cargos without really much of an effect on the moving cargo. So what we learned from our studies here is that stationary cargo clusters act as local traffic jams. These arise, which I didn't show you, they arise because collisions occur in the neuron where anterogradely and retrogradely moving cargo dash with each other. They occur when there are big traffic jams, like a big mitochondria is guaranteed to stop vesicles on either sides of it, okay? And anterogradely moving vesicles cannot blow through a traffic jam. Retrogradely moving vesicles, because of its versatile motor, are able to bypass these jams. And what was very interesting for us is that stationary cargo may be reservoirs from which vesicles can be recruited, if need be, without needing to go back to the cell body upon repeated stimulation. So that's an angle that we would love to explore, and we first need to set up good methodologies to look at that. So how am I with time? I've been going great. I'm fine? Okay, that's great. So I took you on the path where I said in the cell body, you need to make the right cargo complex to be able to recruit the motor, and motors don't accumulate. I told you a little story about the axon. I'll finally tell you, you have the motor cargo complex starting from the cell body, moving in the axon, you come to the synapse. What happens next? So there were 2 major hypotheses in the field, and we were actually driven to these experiments. I've been wanting to answer this question for a very long time, but it was very nice that a screen gave us a set of tools which allowed us to sort of try to bring some kind of answer to this question. So essentially what we had was an allelic series of differential cargo binding with a kinesin 3 motor. So let's just do a simple thought experiment. You have the motor cargo complex, it comes to the synapse, it delivers the cargo, motor is released. What happens to the motor? Does it stick around? And that's, in fact, what many people have proposed, that the motor sticks around, diffuses back to the cell body, and then gets reused again. But I'm going to ask you a very simple question. You have a 1 meter long axon. I mean, why would you want to bother about taking a motor back from the synapse all the way to the cell body to do this job again? Okay? But there have been anecdotal evidences, and certainly mentioned at least in Venn review, where motors, they suggested, essentially from 1 or 2 experiments from Hirokawa's group, that they could be degraded at synapses. So we were able to address this directly, in part driven by the genetics that we had already done. We saw that we asked this directly by using a temperature-sensitive ubiquitination mutant. And when we reduced degradation in the entire animal and we looked at the wild-type motor, we could see that motor levels increased only at synapses, not in any other place, showing clearly that motors that came to the synapses were degraded. So there is an implication of this, and this implication is a set of experiments which we finally was able to do in my own lab. So axonal transport was discovered by Paul Weiss, in part motivated by World War II injuries of people who crashed in airplanes, where he wanted to look at axonal biology, right? So he just cut open various animals, cats, rabbits, and tied a suture around the neuron, sewed the animal up, waited several weeks, opened it up again, and this is a figure from his paper, and he could see flow of cargo from the cell body to the synapse, and from the synapse back to the cell body. And this is really a beautiful assay, which is used to this day, because what it tells you, unlike the movie that I showed you, is that bulk flow of cargo taking place over several days. At any given point of time, any cargo can move bidirectionally. What you're really interested in, if you're interested in understanding the biology of the neuron or the animal, is really over time what cargo ends up at the synapse, right? And I tried all through my postdoc to do these kinds of experiments. Unbelievable. 5 years, I tried everything on the sun, nothing worked. When I came to NCBS, I spoke to Kaustubh, and I said, Kaustubh, the only thing I can think of is now to cut the neuron. And he said, yeah, that's very easy to do. Let's just cut the neuron. It's very easy. And thanks to his encouragement, we went ahead and did this experiment where we cut the axon somewhere either close to the cell body or closer to the synapse. And we looked to see if our model is that motors comes to synapses and then gets degraded, is the motor going to come back? You would predict that it should not. And that's exactly what we see. Very little motor comes back. When we block the degradation pathway so that motors can no longer be chewed up at synapses, we now find that the motor can return to the cell body. We back this up not only by this injury model, but doing bleach recovery experiments where we see the same thing. So what we learned from this last study is that the kinesin 3 motor is degraded at synapses through ubiquitination. Our genetics, where we had the allelic series of differential cargo bindings, strongly suggested to us that this happened, that the motor cargo complex came to the synapse, let go of the cargo, and that eventually led to motor degradation. And why is this important? Why might you want to get rid of the motor at synapses? We think this is really important because if you have the anterograde motor hanging around, it could attach to retrograde cargo, and instead of, if you'd looked at our movies carefully, you will see that retrograde cargos just zip through and go back to the cell body. We see that retrograde cargo would now have a competing anterograde motor on it and drag this detrimental cargo back to the synapse, and work that it did at this, as a postdoc showed that accumulation of retrograde cargo at synapses. And this data comes from multiple systems looking at Drosophila, Erica Bible Saxton, Erica Holzbauer work in mouse. Retrograde cargo accumulation at synapses is really detrimental to the synapse and therefore to the organism. So putting all of these little short stories that I've told you so far, we see that regulation occurs at the cell body, where we think motors are held in pools. When you have the right cargo or the right partner, is a story which I haven't told you about, you can attach the motor to the cargo. The motor cargo complex enters the axon and undergoes processive motion, but when it reaches a traffic jam, a cargo intended for the synapse actually ends up stopping much more. And that might be the way in which you build up pools all along the axon. These pools can release vesicles. We don't really know where they go. We suspect they go towards the synapse from some preliminary experiments. Upon repeated stimulation, so based on cellular needs, you're able to release pools of vesicles from the stationary cargo. We've got a story developing in how vesicles go to synapses, which not. And then once you get your motor cargo to the complex to the synapse, you release your cargo and the motor gets degraded. We're also working on the retrograde pathway. We've developed tools, but it's not ready for prime time yet. So I should tell you who did the work. Jitendra did some of the motor degradation story and did the suppressor screen. Sudeep developed the microfluidics, and all of the imaging, some of Sudeep's movies, Krishna was kind enough to show, were taken by Sudeep and another grad student of mine who's not here, because, Shikha, because showed that motors got degraded at synapses, and he's done the entire M16 story. Kausalya was a former postdoc who worked on the stationary cargo. I have really amazing interdisciplinary collaborators who, partners in crime, Gautam with whom I've developed a model, Kaustu with whom I did the laser axotomy experiments and the microfluidics, and Venkat with whom we continue to work on developing microfluidic devices. We got lots of transgenic lines from Naoki for the LRK story, and the UNC104 GFP and the RAP3 GFP from Mike. These are all my funding sources. Thank you.
Plenty of time for questions.
Oh, I'm glad I finished in time. Okay.
I have a question for all three speakers of the morning, taken together. The C. elegans system has been studied extensively by now, and a lot of how and what questions have been answered about the mechanisms. Are you at a stage where you can begin to ask evolutionary biology questions about the why this system happened?
Kathy? I think it's always the why question. It's very hard to answer. So I think we can try to think of the why question. I mean, people are doing that. I mean, it would not be unreasonable. I mean, other people are better qualified to say that. I can say it from the context of some of this work that we did, that we're very, very interested in the why question. But unless you know the how question, you can't even begin to approach the why questions, which is why we have focused so much of our efforts in first figuring out the cell biology, so we can now begin to link cell biology to behavior and then ask, why does it need to be this way? But that's just our experience. Kathy and May can fill in you.
I have a question for you. So you said the motors, once they've delivered the cargo at the very end of the axon, why would you have it diffuse back? But can you be more precise? Is there a reason it cannot diffuse back?
No. I think so.
Will the accumulation be too large to see any substantial concentration at a cell body that's a meter away?
We had such a hard time publishing that paper because of this. So, so I can give you what I've thought for a really long time. I think there are two classes of cargoes. One should never think about, motors are a means to an end. What the cell really cares about is not the motor. They care about the cargo. And there are different classes of cargo. We also work on mitochondria. The cargo, this cargo is unidirectional. It needs in one place, very far away, very localized. So the kind of regulation, and you make gobs of synaptic vesicles in a neuron. And you make gobs, really huge amounts of the motor. So in this system, I think actually it makes better sense to degrade the motor at the synapses because you're making so much of both. And it's a constantly on system. But if you want a distribution, then you need a completely, a large part of the molecular, the biophysicists who work on motors or the biochemists who work on motors talk about bidirectional motion of cargo. And in that case, when you want to set up distributions, you want to regulate your motor completely differently. So the cargo is what decides how you want to set up your, how you want to set up your motor activity. So there are going to be cases where the motor is going to diffuse back, a motor which has multiple cargoes like kinesin 1, or a motor which wants to set up distributions like kinesins and dyneins. They don't get degraded, I think. So there's going to be a very, it's dependent. It's not as if they cannot diffuse back to the cell body. What I'm saying is it's the cargo which drives whether a particular motor goes back to the cell body or does not. I think this. We have to test it by doing switches. Someday we'll get to it.
I have a related question. So the evidence for the use it or lose it model of motor degradation is a bit indirect. It comes from the ubiquitination experiment. So I was wondering if you could do a more direct experiment by photoconverting
Yes.
motors, for example.
That's next on the plan. That's next on the plan. We need to do that. We need to photoconvert. So we would photoconvert in the touch neuron branch, which is great location, and watch it go to the synapse, and then see what happens. But I want to say one more thing. It's not as if zero degradation occurs in the axon. And those also come from the axotomy experiments. So if you cut off the posterior end of the neuron, which is not attached to the synapse, you can still see a little bit of degradation going on there. That clearly tells you, I mean, to my mind, this use it or lose it is exactly right. But if you fall off the motor along the axonal path too, you could be removed. So I think you don't want a lot of free, a very strong, kinesin 3 is a very strong, very directional, it's a really beautiful motor. And it, you know, it's not like kinesin 1, which is a generalist, it's a specialist, and you probably don't want it around. I don't know, it's a guess. So we're also doing the follow-up experiments to see if the motor is not degraded, what happens to cargo motion of the motion that's coming back from the synapse. So.
Can I ask another question? So in your model, UNC16 is a negative regulator of egress from the Golgi via kinesin 3. So
Yes.
in the UNC16 mutant, is your model that now the synaptic vesicles hop on kinesin 1?
So
Can you test that?
That's a great question. I mean, obviously there must be a balance between the cutting enzyme and the motor. What we find is these large tubes form and move independent of kinesin 1 as well as kinesin 3. Their speeds change a little bit, so we suspect that because these compartments, whatever they are, have many other proteins which have the capacity to non-specifically recruit lots of motors. So Ken Miller has a study as well, and both of us have looked extensively at a large number of motors to see if any of them would block this kind of, you know, egress from the cell body. And he's been looking at lysosomes, and we've been looking at synaptic vesicle proteins, and we haven't found one. So my guess is there's complete change in cargo selectivity, because you're no longer sorting components correctly, so you make all these aberrant large tubes, which we can even see in the EM, and they just non-specifically recruit motors. This is my guess. It's very unsatisfying, but that's what we seem to find.
Just one question. Sorry? Okay. I think, because I haven't worked on this for many years now, so I think the traditional view of during synaptogenesis exercise was the early batch of the vesicles goes to the synapse, and then it contributes a lot. But then afterwards was really the endocytosis, all those events contributes.
Yes.
To the, but in your view, how much of this retrograde antrograde transport really contribute in the vesicle recycling?
That's a beautiful question. So I think you can. That's a beautiful question, May. So basically, I think at synapses, you can of course refill and reuse. You need some turnover taking place. And we still don't understand what drives the turnovers, what has to go, how's the decision made as to what goes back. That is why I think this delivery system, when we did the repeated stimulations, for instance, we don't see it early on because the reserve capacity at the synapse is capable of handling it all. But I think when the animal is getting continuously stimulated, it sort of says, you know, we don't know what the threshold is. We need to find that out. But at some point it says, aha, I really need to make sure I'm going to be okay because I'm reusing this pool so much that we need some turnover taking place. It would be great to get some numbers. We have some good ideas as to how to try it, but we have not started those experiments.
That's what I was thinking when you said
yes.
you can immobilize that, right?
Absolutely.
A specific type of synapses that needs to adapt to different strengths.
Yes, and we'd love to do that. So, you know, we would love to be able to look at other neurons to see if it is next PhD student.
Okay, if there are no more questions, let's thank Sandhya, and we'll take a break.